We have spent the last 6 videos looking at autism as one condition, one diagnosis, one spectrum, one set of criteria. And that framing is clinically useful. But from a research perspective, it creates a serious problem. Here is the problem. Autism is not one condition biologically. It is hundreds, possibly thousands, of different neurological profiles that happen to share enough surface features to receive the same diagnostic label. When researchers try to find a biomarker for autism, a blood test, a brain scan pattern, a genetic signature that reliably identifies autism, they keep running into the same wall. The signal drowns in noise. Because you cannot find a reliable biological marker for a category that contains people with completely different underlying biology. Averaging across that heterogeneity does not reveal the biology. It hides it. This is why, after decades of research, we still have no robust biomarker for autism diagnosis or treatment. Not because the science is failing, because the question has been too broad. The solution, and this is where autism research is genuinely moving right now, is stratification. Instead of studying autism as one thing, researchers are studying well-defined subtypes. Groups of autistic individuals who share a specific genetic variant, a specific brain phenotype, or a specific biological mechanism. When you study a subtype, the noise collapses, the signal becomes visible, and the path toward understanding, and eventually toward targeted treatment, becomes real. Think of it this way. Imagine trying to find the cause of cancer as a single entity. We know now that is the wrong question. Breast cancer, lung cancer, leukemia, same label, completely different biology, completely different treatments. Autism research is going through exactly the same conceptual shift right now. Over the next several videos, I am going to take you through 6 of the most important and well-studied autism subtypes, each one a window into a different neurobiological mechanism. We will look at megalencephaly and microcephaly, what brain size tells us about cortical development. We will look at PTEN autism and SHANK3 autism, what growth pathway failure and synaptic scaffold disruption reveal about how autism emerges at the cellular level. We will look at CHD8 autism, one of the most replicated autism risk genes, and what chromatin remodeling tells us about how a single gene can regulate hundreds of others. We will look at Fragile X syndrome, the most common inherited cause of autism, and the scientific story that generated the first rational drug target in autism research. And we will end with tuberous sclerosis, where the mTOR pathway, cortical development, epilepsy, and autism all converge in one condition, and where the most promising targeted treatments in autism research currently stand. Each of these subtypes is a different entry point into the same underlying question. Why does the autistic brain develop the way it does? And what can we do about it? That series starts next. Follow for more real neuroscience. No tips, no hype, just the science your family deserves to know. Next, megalencephaly, when the brain grows too large, and what that tells us about autism.